All right, in this last part of the unit, in this last video, we're going to take a look at active transport. In active transport, particles, dissolved substances, will be moved against the concentration gradient. Remember, you have a concentration gradient whenever the concentration is different on opposite sides of a semi-permeable membrane. In passive transport, we talked about everything moving from high concentration to low concentration. So, simple diffusion was a dissolved substance moving from a high concentration or a concentrated solution to a low concentration or dilute solution. In passive transport, everything flows from high to low, downhill. Active transport is very much the opposite. It flows uphill. The dissolved substances flow from low concentration to high concentration. And as you can imagine, moving anything uphill requires the input of energy. For example, if you're on a bicycle and you're at the top of a hill, you can just coast down that hill. Let gravity do all the work. Going downhill from high to low doesn't require any energy. But to go uphill, imagine now you're sitting at the bottom of the hill on a bicycle and you need to get to the top. You're going to have to input or use a lot of energy to move that bicycle to the top of the hill. Active transport is uphill. We're going from low concentration to high concentration. This is going to require energy from the cell. The dissolved substances are actively pumped across the cell membrane. This active pumping, this moving them uphill, this moving them from low concentration to high concentration requires energy. It gets that energy from the cell in the form of ATP. You're going to learn a lot about ATP when we do cellular respiration. ATP, in a nutshell, is a cell's energy molecule. You eat glucose and your cells burn glucose to make energy. In a nutshell, again, your body takes the energy that's in the glucose and makes ATP. Then whenever your cell needs energy, it uses the ATP to get that energy. Cells will use this active transport process to either concentrate a specific substance inside a cell or to remove waste products from the cell. We're going to look at three processes as examples of active transport. We're going to look at sodium-potassium pumps. We're going to look at exocytosis and endocytosis. So like we had three forms of passive transport, simple diffusion, facilitated diffusion, and osmosis, we're going to have three forms of active transport to learn: sodium-potassium pumps, exocytosis, and endocytosis. So starting with the sodium-potassium pumps. It is another form of transport that is run by or facilitated by a protein. Except what we're going to be doing here is moving things against the concentration gradient. Here's how it works. The initial state of our protein is in a cone shape with its opening facing towards the inside of the cell. As this opening is facing towards the inside of the cell, sodium ions can move into the protein and bond to one of three sites. Each protein can bond with up to 3 of these sodium ions. And again, this occurs when the protein is opened up towards the inside of the cell. When it's full of sodium, an ATP will attach to it, giving that protein energy and causing it to change shape. The cone flips. It's closed now towards the inside of the cell, but open towards the outside. In this state, those 3 sodiums are released and free to exit the cell and move to the extracellular environment on the outside. Once those sodiums are gone, potassium ions can move into the pump. And the pump is able to bond with 2 potassiums. The pump bonds to 3 sodiums and moves those 3 sodiums out of the cell. Then it will bond with 2 potassiums. Once it's bonded with those 2 potassiums, the pump will flip its orientation again, open up towards the inside of the cell, where it will release those potassiums to the internal environment. So 3 sodiums enter the sodium-potassium pump from the inside of the cell. ATP gives that pump energy, causes it to change shape, open up to the outside of the cell. The 3 sodiums are released. They go to the extracellular or outside environment. 2 potassiums come in, bond to the pump. The pump changes shape, opens back up to the inside of the cell, where those potassiums are released to the intracellular environment. So you see 3 notches over here. Those are the 3 notches for the 3 sodiums that it can move out. You see 2 notches on the other side. Those 2 notches are for 2 potassiums that it can move then in. This pump does not work without ATP. Here's a little animation. The 3 sodiums move in and bond to the pump. ATP comes along, attaches to the pump, gives it the energy it needs. It changes shape, opening up to the outside world where the sodiums are released. 2 potassiums can then move in and bond to the pump. Pump changes shape, opens up to the inside of the cell, releasing those 2 potassiums back to the inside. Again, 3 sodiums go in. Energy from the ATP. 3 sodiums go out. 2 potassiums take their place. Changes shape again. Releases those 2 potassiums to the inside of the cell. Sodium-potassium pumps obviously work with sodium-potassium ions. These are very small particles. There are large substances, however, that can't make it through the cell membranes at all, be it through the membrane itself or through any of the other passive means like an ion channel or a carrier protein. They're simply too big to fit any of these things. Some of these things are lipids, for example. They're going to have to move across the cell membrane in a vesicle. If you remember from the last unit, a vesicle is one of these little packets that is used to transport substances either internally in the cell or into or out of it. Endocytosis is when a vesicle is used to move something into the cell. Endocytosis, enter. They both start with EN. Endocytosis, endo means enter. The vesicle attaches to the cell membrane, wrapped up by it, and then opens up to the inside of the cell once it's into the cell. Again, using this vesicle, using what is a phospholipid bilayer to attach to and pass through the cell's own phospholipid bilayer. Exocytosis is when a vesicle is used to remove something very big from the cell. Again, something like a lipid that's too big to get through one of the carrier proteins. Exocytosis starts with EX, so does the word exit. That'll help you remember that exocytosis is when something exits the cell in a vesicle. Again, what's going to happen is the vesicle is going to attach to the phospholipid bilayer that is the cell membrane, open up, and release those large particles to the outside world. The formation of vesicles requires energy from the cell. So both endocytosis and exocytosis are forms of active transport. That'll do it. That is the end of the unit and everything that will be on your next unit test.